Okay, today I want to talk about the digestion of carbohydrates. One of the things that I like about biochemistry as an inorganic chemist is there's so many things about our day-to-day life that can help to be explained by biochemistry. And there's a lot of things that we maybe understand intuitively, like we eat food, and then that food allows us to have energy to do things. And there's something about the breaking down of that food that allows us to create new things. And that's sort of all part of this internal process that we do every day, but maybe don't give a lot of thought about the details to. And the cool thing about biochemistry is it allows you to fill in some of those details in a pretty accessible way. So that's kind of fun, I think. I think it's fun. So let's talk about carbohydrates. So carbohydrates are sugars. These can range anywhere between simple sugars like the monosaccharides, which are the monomers that make up these gigantic large chain polymers, these complex carbohydrates, and mostly what we eat when we're thinking about foodstuffs are larger carbohydrates, complex carbohydrates. So we're talking about things like starch, right, in the form of either amylose or amylopectin. We have cellulose, which is dietary fiber, that comes from plants as well. We have glycogen, which comes from proteins, meats, animal-type products. All of those things, when we eat them, are these long chains of monosaccharides. In the case of most of those kinds of things, we're talking glucose. So these long chains of glucose molecules that need to be broken down in order for us to use that glucose, because that glucose is going to be the primary driving force between all metabolism. And that gets us into things like the breakdown of glucose gives us pyruvate, and pyruvate gives us the acetyl-CoA, and then acetyl-CoA can give us the start of the citric acid cycle, and the citric acid cycle goes through, and then you end up in the electron transport chain, and we get energy. So when we say, well, carbohydrates and sugars give us energy, they do, but it takes quite a bit of process in order to do that. So try to keep up with my art skills here. We have a carbohydrate. I was thinking muffins because there's a froyo place here in my little town that does Muffin Mondays. So I was thinking about muffins yesterday. And so we eat a muffin, and digestion is the entire process. We usually think about digestion as what happens in the stomach. But on a more scientific kind of basis, digestion is the catabolism, so the breaking down from larger into smaller pieces of food molecules. So the stuff that's in our food, we're breaking down into its component pieces, and then we can use those component pieces to either get energy, we can build them back up in order to make molecules and proteins and enzymes that we need to do other things. So digestion is very important, but it's kind of the generalized process. Digestion, catabolism, metabolism, they all have slightly different but interconnected meanings. So when we're talking about digestion, usually we're focused on the physical mechanisms of it, right? So I put the muffin in my mouth. That's not actually what I look like. I have more of a neck, actually, than that. That looks like a brontosaurus head. So if my brontosaurus head here eats the muffin, then the first step in digestion is here in the mouth, right? So in the mouth, we have kind of the salivary glands, and the salivary glands excrete alpha amylase. That's an enzyme. We can tell that it's an enzyme because of the ACE ending. That ACE means that it acts on something, and this alpha amylase speeds up the hydrolysis of all of these larger carbohydrates. So starting right there in the mouth, in the saliva, we have all of the stuff that's in there. So all these, starch, and all of our, glycogen if we're eating meat or animal products, or some of the larger type carbohydrates. So, disaccharides, polysaccharides. So all of these start off as coming into our mouth, and they can start being broken down by this enzyme that's in our saliva, this alpha amylase. And the alpha amylase acts only on alpha of our, when we're thinking about isomers here, the alpha isomers of our different starches and such. So when we think about the connections, the glycosidic bonds between our sugars, then if it has an alpha linkage, we'll put the term there, alpha glycosidic bonds, then that's what it works on. And so it works on those to cleave them apart. That's hydrolysis. Hydrolysis. So we're using water and this enzyme to break things apart. And this happens right away in your mouth. So in our mouth, then the saliva is acting on the food. And of course, we don't keep the food in our mouth for very long. At least I hope not. That would be a little bit disturbing. But some people need to chew more than others, right? I don't chew my food enough. And now, actually, the more I learn about biochemistry, I think the more I need to chew. That's my big takeaway from all this. Okay, so the saliva in your mouth starts to work on this, but it also works all the way down the esophagus, right? So our esophagus leads down to our stomach. So we have our stomach down here. And it's all kind of connected by a tube. It's not closed off. It's obviously connected to other things. Obviously not to scale, otherwise it has the shortest torso in the history of the world. My poor little brontosaurus here. Okay, so it gets into our stomach, just for clarity's sake. So all of the thing that's happening from the mouth to the stomach is the saliva is working on it all the way down the track, right? So from a physical standpoint, we chew things up. When we chew things, that breaks it into physically smaller pieces, right? That's what the teeth are for. So we masticate things, we break them into smaller pieces. Those smaller pieces then are processed by this alpha amylase, and that works all the way down until you get to the stomach. So all of these things that are found in food are then broken down into smaller bits. So now we're just broken down into our polysaccharides, right? If we're starch or glycogen, we're broken into, if we're polysaccharides, maybe we're broken into things like lactose, maltose, some of the other disaccharides, and some of these will start getting cleaved into smaller bits as well. So some of them will end up as monosaccharides as well, but that's usually a process that happens a little bit further along. It depends on what type of sugars they are that we're talking about in your foods. So the mouth, the saliva works all the way down the esophagus. Now in the stomach, we know that the stomach acid is highly, acidic, hence the name. So we have a pH of around 2 in our stomach, 2-ish. And not a lot happens here, and this actually suppresses the alpha amylase. Not much going on. So because of the difference in pH, we know that pH has an influence on how these things break down, right? And how enzymes work, because there's different factors that can inhibit or promote enzyme activity, and pH is one of those. So alpha amylase does not work well in the pH conditions of the stomach. So there's not a lot that happens for the digestion of carbohydrates in your stomach, which is sort of counterintuitive, because when we think of digestion, again, from kind of a layperson standpoint, we think, oh, well, it's what happens in the stomach. But really for carbohydrates, it's all about the saliva. Now, the next step is also important. So things are getting processed in the stomach. Again, not a lot going on. Now our pancreas, it's a good pancreas there, is going to start secreting this alpha amylase into our small intestine. So we have our small intestine here. We're going to wrap around there into our stomach. So now our pancreas is going to secrete this same alpha amylase, this time from the pancreas. And so now that alpha amylase into the small intestine is going to start breaking these things down again. So stomach is sort of an intermediary step. It's sort of hanging out there. It's getting broken down again physically by the acid, potentially, but not by enzyme activity, because the enzymes were not present or not able to work in that condition. So now the pancreas excretes that alpha amylase, and in our small intestines here, then we start getting more action that happens. So when we're thinking about breaking these larger carbohydrates down, so these polysaccharides, these disaccharides, then in the small intestine, this is really where all of that happens. So all these polysaccharides and disaccharides, we're getting down to the monosaccharide level. So it's essentially cleaving everything into its component pieces. So let's remind ourselves kind of what these large molecules look like, because there's a few different ones that I've mentioned, and I just want to remind you of the structure of them. So amylose and amylopectin are both considered to be starches. So these are from plants. Starch. Same with amylopectin, plant. Starch. Okay. So if we think about these, they're just long chains of glucose. I have these in the chair conformation in kind of a two-dimensional way. So we have our glucoses here that are aligned. So we have a 1, 4. These guys are both in the alpha conformation. So we have alpha 1, 4 linkages here. And because of this chair conformation and the alpha 1, 4 linkages, this allows for this kind of helical structure of amylose. So it's a helix. Whereas amylopectin, if we kind of have these as basis for comparison here, amylopectin is branched. So we have these alpha 1, 4 linkages here, alpha 1, 4 between these guys, but then there's also branches. There's some 1, 6 alpha linkages here. So here's a 1, 6. Okay. And again, you can't see this very well because of my horrible drawing skills and my two-dimensionality, but this would be alpha as well, so alpha 1, 6. So we have these alpha 1, 6 branches off of the alpha 1, 4 chains, and that allows for kind of this branch structure of the amylopectin. Branched. The comparable molecule in animals is glycogen. So glycogen also has these branches with the same structure for the same reasons. They're just created in different ways, right? Plants, of course, are putting together these glucoses based on, their photosynthetic processes, and then all that gets built in that way, whereas glycogen in animals comes from, building muscle tissue and all that kind of stuff. That's where we store a lot of that. So these processes are similar because they create similar looking molecules. Remember, structure, function, that all works together. Okay, so the alpha amylase then, this enzyme that comes in, is going to come in and cleave these bonds and break these guys apart at these alpha linkages. So they could break these guys apart, break these guys apart. Now we're getting into component pieces. So now I have a little part of a branch here, part of a branch here. And if I keep breaking them down further and further and further, ultimately I end up with glucose molecules. And that's kind of the name of the game here, is we're trying to get to glucose. Now, just as a side note, because I think it's sort of interesting to compare the structures. So we have, again, amylose and amylopectin with its distinctive structure. Cellulose is also created from plants. Cellulose is the primary component in cell walls, so it has more of a structural type function as opposed to an energy storage type function, which we see in starches. So cellulose for plants, so this is kind of, let's go and just label it structure, in our plant walls, comes about because this linkage now isn't an alpha linkage, it's a beta linkage. So it's beta 1, 4. And then this would be a beta 1, 4, beta 1, 4, all the way up. So we have these kind of long chains of glucose that would give you kind of strands, essentially, structural strands for your cellulose. And these are not digestible by humans then because we don't have beta amylase, essentially. So we have alpha amylase in all of our digestion process, but we don't have a way of breaking these down. So dietary fibers like these will just come kind of straight through your whole digestion process. It helps to physically clean things out, but it's not something that we can break things down into its component pieces and digest in the scientific term of breaking apart these molecules into their component pieces. Okay, so if we go back to our diagram then, when we start breaking those down into these smaller and smaller pieces, so from the large ones into polysaccharides, so kind of complex carbohydrates to polysaccharides, polysaccharides to disaccharides, disaccharides to monosaccharides, then we get down to individual glucose molecules. And those individual glucose molecules are going to be used for then our metabolism process. And Sorry, got distracted there. If you have any questions on that, please don't hesitate to reach out. Digestion is kind of a cool thing, and it's, again, all about, building up of molecules after breaking them all down, and the breaking them all down happens through digestion.